This is the first part of the lecture on the vessels. We're going to start with vessel anatomy. This is covered quite well in your book. The three basic vessel types are arteries, capillaries, and veins, and we'll be looking at the layers and where they're found and what the types are. So we'll start with the arteries. There are three types of arteries. The first two are shown on this slide, elastic arteries and muscular arteries. Elastic arteries are sometimes referred to as the conducting arteries or the great arteries. This includes the aorta and its major branches, and likewise the pulmonary trunk to some degree, but the pressure in the pulmonary trunk is much, much less than the pressure in the systemic arteries. So in the elastic arteries, you'll notice you have a very thick tunica media, but it's mostly elastic tissue with much less smooth muscle. You'll also see that the smooth muscle fibers tend to run along the the longitudinal axis of the vessel, so that when they contract, they're not going to constrict the vessel. Elastic arteries really don't constrict very much. What they do is they expand with each beat of the heart. Remember that little 1 inch of ascending aorta has to accept 70 milliliters of blood in like less than half a second, even at rest. So they've got to expand very quickly, like a balloon being blown up, and then as they contract, they just send that blood on down their branches. This also, of course, shows the three basic layers of a blood vessel. The tunica intima, the innermost lining, is endothelium, which is a simple squamous epithelium and just a very thin layer of loose connective tissue. And then the tunica media is some varying amount of elastic tissue and smooth muscle. The tunica adventitia can be quite thick. It is another layer of loose connective tissue, and depending on how big the vessel is, well, those vessel walls are so thick they may need their own blood supply. So the blood vessels that supply the blood vessels are in the tunica adventitia. If you look at the lower figure, you see a typical muscular arteries. These are often also called the distributing arteries. Beyond the great arteries, these are going to be most of the named arteries that you learn. The brachial arteries on out to the limbs, likewise femoral on out to the lower limbs, and the vessels that supply individual organs are going to be muscular arteries. Contrast the tunica media in the muscular artery and the elastic artery, and you see, yes, there's still elastic tissue. In fact, there's like two distinct sheets of elastic connective tissue, an outer one and an inner one. But sandwiched between them is a layer of smooth muscle, and notice that smooth muscle is circularly arranged around the vessel's longitudinal axis, so that when that muscle contracts, it will constrict the vessel. The pressure in muscular arteries is so high that you cannot constrict them all the way closed, but they're going to vasoconstrict always to some degree. Maybe you could consider it about a halfway vasoconstriction, so they could vasoconstrict more or less depending on the needs of the body. So if you're trying to reduce blood flow, say, to the digestive organs when you're running the track, you can vasoconstrict those vessels more, whereas the vessels that are supplying the musculature that you're actively exercising, those vessels will dilate. Notice, again, you still have the consistency of the tunica adventitia and the tunica intima. Those are pretty well standard. Now, in this diagram, it looks like the inner layer of elastic connective tissue is considered part of the tunica intima. You know what? That's just really not a major issue. Let's look at the third kind. It's an arteriole, and in the wall of an arterioles are considered arteries, you're going to see very little elastic tissue and a pretty significant smooth muscle that can vasoconstrict. Arterioles can actually vasoconstrict completely to cut off the blood flow to a particular capillary bed. The arterioles lead into capillary beds, so every capillary bed has to have an arteriole leading in and a venule leading out. As we'll see in the diagram, there is a point at which the arteriole can connect directly to the venule. That's called an anastomosis. Now, ana means apart, and stoma means opening. So anastomosis means just a direct connection, a thoroughfare channel, we call it, that will bypass the true capillaries. To identify a true capillary, we always look for a precapillary sphincter. the precapillary sphincter is simply the last little smooth muscle cell that encircles the entry to a capillary. Again, it can completely close so that no blood can flow through specific capillaries. So we'll talk more about regulation later. So here in this diagram, you can see the arteriole with just individual layers of smooth muscle encircling it. They're sort of scattered along the wall of the arteriole. Arteriolar walls have an endothelium. All blood vessels do. And just that thin layer of smooth connective tissue, and yes, they're going to have a scattered layer of tunica adventitia around the outside. Look at the thoroughfare channel, and you can see that that bypasses the capillaries and dumps directly into the venules. And each precapillary sphincter marks the opening of an individual capillary. In this diagram, it looks like capillaries branch off each other. I don't know if that's true or not. Doesn't really matter. Capillaries are going to lead off of arterioles and drain into venules. And if you want to control the flow of blood through an individual capillary, you close the precapillary sphincters individually. If you want to control the flow of blood to the entire capillary network, you close contract the smooth muscle cells in the arteriole and close off the complete arteriole. All right, so we're going to focus next on the types of capillaries. We have three basic types. All of them have consistently the same layers. The endothelium, which again is a simple squamous epithelium, very thin basement membrane. Those epithelial cells are connected to each other by varying consistencies of tight junctions and desmosomes. And I know you remember tight junctions actually seal the boundary between epithelial cells so no fluid can leak out. Desmosomes just sort of stitch the cells together so they don't separate from each other. Our three types of capillaries are called continuous capillaries, fenestrated capillaries, and sinusoids. Continuous capillaries are just what they sound like. It's just a typical capillary that is the lumen of the capillary is continuously lined with these endothelial cells. But in most continuous capillaries, there are gaps between the cells. We call these gaps intercellular clefts, and that's just where there aren't tight junctions. So fluid can, in fact, leak out of the capillary and into the tissue spaces. What leaks out is the plasma without its plasma protein. So those intercellular clefts are narrow enough to prevent plasma proteins from leaking out and becoming part of the tissue fluid. And we'll study that later as we get into the dynamics of how capillaries do nutrient and waste exchange. Not all continuous capillaries are leaky. Wherever there's a capillary barrier, such as the blood-brain barrier, there's also a blood-thymus barrier and a blood-testis barrier. There are continuous tight junctions that prevent the leaking of any fluid, typically in those specific organs. And so I have a special term for those. I call those tight continuous capillaries. That's just a term I made up to help you distinguish them. So you have tight continuous capillaries in nerve tissue. You have tight continuous capillaries in the seminiferous tubules where sperm are formed in the testes, and you have it in the thymus to prevent the leakage of proteins that would engender an immune response to your own proteins. That would be bad. The second kind, the fenestrated capillaries, have all the things a typical continuous capillary has, like intercellular clefts and, of course, the desmosomes. But the endothelial cells themselves have lots of little tiny, we call them pores, but for most of the fenestrated capillaries, it's not really an opening. It's just a thin spot where the endothelial plasma membrane on the inside merges with that on the outside, and these little pores are full of carriers for things like glucose so that they can be rapidly transported through the capillary wall. Now, in the kidneys, there are special fenestrated capillaries where the pores really are open holes and stuff can just flow pretty freely. Again, not the proteins. We don't let proteins leak out of either continuous or fenestrated capillaries. The third kind is called the sinusoids. Sinusoids, they're capillaries in the sense that they provide the functions of capillaries in a typical tissue, but you have an unusually wide diameter. So the diameter of a typical capillary is just wide enough to let red blood cells flow through. So if a red blood cell has a diameter of about 8 micrometers, well, a typical capillary diameter is about maybe 10 micrometers at the most. Sinusoids are much wider than that. And one of the things that does is it slows down the flow of blood. Sinusoids also typically have hairpin turns in them. And in these spots, only the young, flexible red blood cells can bend and squeeze through. So any old red blood cells tend to get caught here. And that is where we're going to remove old red blood cells. So you already know where that happens. It's in the spleen and the liver. So you have sinusoids in liver, you have them in spleen, and you also have them in bone marrow because red blood cells have to get into the blood supply in the bone marrow. And it takes the wide openings of these sinusoids to allow that to happen. We'll see a diagram of that in just a minute, so we'll come back to that. So here's a typical capillary, a continuous capillary, and you can see the endothelial cells just kind of line up against each other. And you can't see because it's not diagrammed, but wherever there's a boundary between two endothelial cells, there will be those intercellular clefts that just allow fluid to leak out of them. Just think of a leaky garden hose. And we have a comparison to the diameter of a red blood cell, so you can see that the diameter of the capillary is just big enough to let red blood cells go through. Single file. So our continuous capillaries are found in most just run-of-the-mill body tissues, muscles, connective tissue, and of course that tight continuous type is found in nerve tissue and thymus and testis. Fenestrated, wherever the load of diffusion is really high, particularly for things like glucose and other small organic molecules, so in the intestinal epithelium, in the kidneys, and some of your endocrine glands, and of course that would be the ones that are secreting the peptide hormones. Our sinusoids are found wherever you've got either large particles or really high volume that has to get through liver, spleen, bone marrow, and some lymphatic organs. So here is a fenestrated capillary. You see the little black spots indicating the fenestrations. You see the purple area indicating the intercellular clefts. You've got the basement membrane kind of peeled back so you can see the bare naked surface of the endothelial cells. And again, the diameter here I think is a little too wide, but we're not going to quibble over that. The diameter of a fenestrated capillary is probably about the same as a typical capillary. And then here's a gorgeous illustration of a sinusoid in the liver. Now, get your bearings. Find where the liver cells, the hepatocytes, are labeled. Those are the ones that look like simple cuboidal epithelium, because they are, with microvilli on the surface adjacent to where the capillary is, the sinusoid is. So as you're looking at what's labeled the endothelial cells, you see they're not even really connected to each other. There are big gaps between them. Sinusoids are often also called discontinuous capillaries. And look at how wide it is. You've got just like room for many red blood cells across to get through a sinusoid. Notice also at the top you have a macrophage labeled. These are the liver macrophages. They're also called Kupffer cells. And these are the ones that are, of course, going to eat old red blood cells and remove them from circulation. They're also going to help with other liver functions. So just as an introduction to the histology of the liver, you have two basic kinds of liver cells. You have hepatocytes, which are the epithelial cells. They're considered glandular epithelium. And you have the liver macrophages. All right, so our third kind of vessel is the veins. There are three categories of veins: venules, veins, and sinuses. Venules are the smallest ones. Remember, those are the ones that drain the capillary beds. They have really nothing more than an endothelium and maybe a thin layer of connective tissue. There's no tunica media in venules. The veins have a significant tunica media. The difference between small, medium, and large veins really is just the diameter. Small veins are going to be draining areas within an organ. Medium veins are going to drain individual organs, and then the large veins are the veins that drain multiple organs, like the superior vena cava and the inferior vena cava and their major branches. Venous sinuses are particular structures that are part of the venous circulation because they drain blood back to the heart. A sinus, in the terms of a venous sinus, is unusual in that it is the connective tissue associated with an organ lined by endothelium. No tunica media, therefore no vasoconstriction. And there are only two that we pay attention to. The coronary sinus, which you already learned on the back of the heart, is endothelium lining the visceral pericardium of the heart. They're on the back of the heart. And the dural sinuses are in the dura mater of the brain. You learned one last semester. That was the superior sagittal sinus. That's the one that cerebrospinal fluid drains into. You might want to go back and look at that. There are dural sinuses wherever there's a major fissure on the surface of the brain. So you have a transverse one between the cerebellum and the cerebrum, and then there's some others, and you don't need to know their individual names other than the one you already knew, which was the superior sagittal. The dural sinuses all drain into the jugular veins that are going to drain the head. Some of the veins have valves. Particularly the medium-sized veins in the limbs have valves. Some of the veins don't have any valves. The job of the valve, of course, is to prevent the backflow of blood. So you're going to see them mostly below the heart, not so much above the heart, because only below the heart do you tend to have problems with fighting gravity to get blood flow back up to the heart. So in the limbs, definitely there are valves. When you get varicose veins, when someone gets varicose veins, those varicosities form because the vein wall is so thin, it tends to get stretched out. And if the valve flaps fail to meet in the middle because the valve is too stretched, we call that an incompetent valve. And that puts twice as much blood on the valve below, which then tends to stretch out and become incompetent. So you have this sort of domino effect of failure of the valves, and then the veins become distended and protrude through the surface of the skin. So it's mostly those superficial veins that are going to get varicosities. And you see that typically in people who stand on their feet all day for their job, kind of like nurses. Pregnant women, because the presence of the fetus tends to block the flow of veins, of blood through the veins. And in some other disorders where blood flow is blocked. Okay, so here you see some diagrams of the veins. The medium and large veins have, of course, a tunica media, tunica adventitia, tunica intima. The tunica media does have some elastic connective tissue in it, and the smooth muscle is the primary player there. I see that they have the smooth muscle arranged longitudinally. I'm going to tell you that these things can vasoconstrict, but when they do, they tend to force the blood back towards the heart. And then the lower figure shows you the structure of the venous valves. All right, so we're going to end this part of the lecture with looking at the distribution of blood flow. I'm going to show you a pie chart, and it's going to look like blood is kind of sitting in the vessel. So I want to remind you the blood has to constantly be flowing. But blood flows faster through arteries than it does through veins, and so veins tend to hold more blood or contain more blood than arteries do. There's just more volume within the spaces of veins than there is within the arteries. So if I want to increase the flow of blood back to the heart, all I have to do is constrict my veins, and that's going to increase venous return to the heart, which is going to allow the blood to flow more quickly to the arteries and increase cardiac output. So here's our pie chart. About 18% of the blood is in the pulmonary circulation. These are resting values. The heart holds about 12%. Remember that our total is about 5 liters of blood. Arteries only 11% compared to the veins not holding, but blood, about 54% of the blood volume flowing through veins at rest. And then the capillaries, which is where the action is, that's where nutrient waste exchange occurs, only hold about 5% of the blood at rest, and that means the blood's flowing very quickly through capillaries. So this is distribution of blood as a percentage of the total 5 liters. As we get into the physics of circulation, we'll look at how fast the blood is flowing and what regulates how fast it flows. All right, so we'll start with pressure, resistance, and flow in part 2.